In this section, I have already described two specific examples of new metabolite signals during fasting that have been discovered recently, alanine and glycogen signals. The last topic is about unknown signals during fasting, which suppress SREBP1 under a fasting condition. As previously mentioned, SREBP1 is a transcription factor that belongs to the basic helix loop helix-leucine zipper family and is considered to be controlling the transcriptional regulation of fatty acid synthesis. They bind to DNA as a homodimer as shown here. As shown here, SREBP1 itself is suppressed by fasting, and induced by refeeding at the mRNA level. We previously tried to find the upstream transcription factor that activates SREBP1 gene promoter, and identified LXR. LXR, or liver X receptor, is a member of the nuclear receptor family of transcription factors. LXR forms a heterodimer with the obligate partner RXR, or retinoid X receptor. The LXR and RXR heterodimer can be activated with either an LXR agonist oxysterols or a RXR agonist. So, the involvement of LXR-RXR has been clarified. Then, what regulates LXR-RXR? By ligands? Or by cofactors? That's the question. But, how LXR regulates SREBP1 gene transcription has not been answered until we have recently solved this puzzle through our new approaches. The main difficulty was that fasting and refeeding response observed in the in vivo setting cannot be simulated in cultured cells. Even superphysiologically high doses of glucose and insulin cannot fully induce sufficient response in the in vitro setting, as shown here. Moreover, as we reported before, insulin is not essential for the fasting and refeeding responses, because full induction can be seen in STZ-treated insulin-deficient mice, as shown here. Also, it is published in Cell Metabolism that liver-specific insulin receptor knockout mice showed no significant differences in fasting and refeeding responses of lipogenic genes including SREBP1 gene expression. So, there are difficulties in promoter analysis of fasting and refeeding responses in cultured cells. This is the reason why we tried to perform promoter analysis in the in vivo setting. This is the new approaches we chose to get over the difficulties, in vivo ad-Luc analytical system and TFEL skin method. First, let me explain our in vivo ad-Luc analytical system. In our in vivo ad-Luc analytical system, the promoter or enhancer region of interest is cloned and constructed into adenovirus vector with a luciferase gene, and the adenovirus is injected intravenously into mouse liver. This allows the promoter or enhancer activity of the DNA region to be evaluated under physiological conditions in living animals. As a quantification method of luciferase reporter, the luminescence from the liver is measured under anesthesia using in vivo imaging system. This photo is a representative one showing that refed mice have 20 times stronger luciferase activity than fasted animals. A series of deletion analyses of the promoter region was performed, and the responsible element was found to be located within -250 to -150 BP upstream of the transcription start site. This result was confirmed by experiments using another series of adenovirus constructs encoding different partial sequences of the SREBP1 promoter to estimate the enhancer activity. Intriguingly, the two LXRs alone were not sufficient for nutritional regulation, although they exhibited a complete response to an LXR ligand. This suggests that nutritional regulation is not brought about by changes in the ligand concentration. Further scrutiny clarified that a cis element flanking the LXRE besides the two LXREs is indispensable for nutritional regulation. So, we designated this element as the nutritional regulatory element, or NuRE for short. From the data shown here, NuRE was shown to exert a suppressive effect on the SREBP1 promoter in a fasting state. Consistent with this finding, an electromobility shift assay showed a fasting-specific band. It suggests that some fasting-induced transcription factor in liver nuclei that binds to NuRE may be involved in the suppression of SREBP1 promoter activity during fasting. So, how can we identify what this transcription factor is? For this purpose, we developed a tool we call TFEL, or Transcription Factor Expression Library. TFEL scan is an expression cloning method using this TFEL library. Using this method, we can identify what transcription factor is functionally binding to a cis element of interest. TFEL was constructed from phantom clones by subcloning excised DNA fragments into pcDNA 3.1 expression vector 1 by 1. It is composed of about 1,600 clones. It took 3 years. Now we can screen individual transcription factors that have suppressive effects against the NuRE plus LXRE, in the presence of co-transfected LXR-RXR. After 3 steps of screening, 2 transcription factors, KLF15 and KLF4, were identified as candidates. Among all KLFs, only KLF15 and KLF4 suppress the NuRE plus LXRE in an NuRE-dependent manner. Next we examine the KLF15 and KLF4 expression levels in the liver. KLF4 was not detectable in the liver, while KLF15 was detected in the liver and was markedly increased during fasting. Accordingly, the nuclear protein detected by the electromobility shift assay was identified as KLF15, based on the disappearance of the band when adding anti-KLF15 antibody. Next, we examine the influence of the forced expression of KLF15. When KLF15 adenovirus was transduced into refed mouse liver and the KLF15 expression was elevated to the fasting level despite the fed state, the SREBP1 promoter activity was completely repressed to the fasting level. Conversely, when we evaluated the contribution of KLF15 under the fasting condition by knockdown, the promoter activity as well as the expressions of SREBP1 and consequently fast genes were markedly elevated despite the fasting condition. Next, to elucidate the molecular mechanism of KLF15 suppression against LXR-RXR, the molecular interaction between KLF15 and LXR-RXR was assessed using co-immunoprecipitation experiments. Consequently, KLF15 and LXR-RXR were shown to form a complex in cultured cells and in vivo liver nuclei. From these results, we concluded that the fasting-induced transcription factor KLF15, a key regulator of gluconeogenesis, forms a complex with LXR-RXR specifically on the SREBP1 promoter. This complex recruits the RIP140 co-repressor instead of SRC1 co-activator, resulting in repressing the transcription of SREBP1 and thereby downstream lipogenic enzyme genes during fasting. And the next question is, what induces KLF15 expression during fasting? This question is currently not answered. KLF15 also plays a pivotal role in amino acid metabolism, especially gluconeogenesis from glucogenic amino acids such as alanine. So, we are speculating that the KLF15 inducer might be related to amino acid metabolites. This slide shows how KLF15 is involved in amino acid metabolism. KLF15 expression in the liver is induced when animals are fed high-protein diet, and this response leads to ALT and OTC gene transcription, which promotes gluconeogenesis and ornithine cycle pathways. Accordingly, KLF15 knockout mice exhibited hypoglycemia and hyperammonemia. But, what induces KLF15 expression in this context is also unknown, and is the next big question. As we have seen so far, nutrients also function as information carriers, which is thought to be partly responsible for maintaining the homeostasis of nutrient metabolism. There are still many unknowns about these mechanisms, and nutrigenomics continues to be a hot research area. Our research is mainly supported by the Ministry of Education, Culture, Sports, Science and Technology Grant and Aid Project. This scientific research on innovative areas was organized by Professor Fukamizu at Terra Center. Thank you for your attention.